sweet pea is a flowering plant in the genus Lathyrus in the family Fabaceae, native to Sicily, southern Italy and the Aegean Islands. It is an annual climbing plant, growing to a height of 1–2 meters, where suitable support is available. The leaves are pinnate with 2 leaflets and a terminal tendril, which twines around supporting plants and structures helping the sweet pea to climb. The flowers are purple, 2 to 3.5 centimeters broad, in the wild plant, larger and very variable in color in the many cultivars. The annual species, L. odoratus, may be confused with the everlasting pea, L. latifolius, a perennial. Horticultural development. Henry Eckford, a Scottish nurseryman, crossbred and developed the sweet pea, turning it from a rather insignificant, if sweetly scented flower, into the floral sensation of the late Victorian era. His initial success and recognition came while serving as head gardener for the Earl of Radnor, raising new cultivars of pelargoniums and dahlias. In 1870 he went to work for one Dr. Sankey of Sandywell near Gloucester. A member of the Royal Horticultural Society, he was awarded a first-class certificate in 1882 for introducing the sweet pea cultivar bronze prince, marking the start of association with the flower. In 1888 he set up his development and trial fields for sweet peas in Wim in Shropshire. By 1901, he had introduced a total of 115 cultivars, out 264 cultivars grown at the time. Eckford was presented with the RHS Victoria Medal of Honour for his work. He died in 1906 but his work was continued, for a time at least, by his son John Eckford. More recently, the association between the sweet pea, the Eckfords and Wem has been highlighted again. In the late 1980s, the Sweet Pea Society of Wem started an annual show and the town has again taken the flower to its heart. Many of the street signs now carry a sweet pea motif and an area of the town is known as Eckford Park. There is also a cultivar Dorothy Eckford, named after a family member. Cultivation. Sweet peas have been cultivated since the 17th century and a vast number of cultivars are commercially available. They are grown for their flower color, and for their intense unique fragrance. They are grown by gardeners for private consumption or for exhibition, and in the floristry trade. The large, pea-shaped seeds are sown in cold frames in spring or autumn. The seeds benefit from pre-soaking or chipping with a sharp blade. The plants are also available later in the season, as young plants or plugs. They are grown-up canes, with the new shoots being regularly pinched out to promote a bushy habit and higher flower yields. Plants typically reach heights of 1–2 m, with the flowers appearing in midsummer and continuing for many weeks if regularly dead-headed. Over 50 cultivars have gained the Royal Horticultural Society's Award of Garden Merit. See List of AGM sweet peas, pests and diseases. The sweet pea plant does suffer from some pests, the most common being aphids. These insects suck the sap out of the plants, reducing growth. Mosaic virus is spread by green fly, causing yellowing of leaves, distortion of new shoots, and inhibited flowering. A pest called the pollen beetle, which is small, shiny and black, eats the pollen and disfigures the flowers. Other pests include caterpillars, thrips, slugs and snails. Another problem is mildew. This is a white powdery coating that covers the leaves and slows down growth. The sweet pea is also susceptible to ethylene in quantities produced by senescing plants. Because of this, growers are encouraged to plant sweet peas away from fruit trees among other plants prone to early dieback or senescence. Toxicity. Unlike the edible pea, there is evidence that seeds of members of the genus Lathyrus are toxic if ingested in quantity. A related species, Lathyrus sativus, is grown for human consumption but when it forms a major part of the diet it causes symptoms of toxicity called lathyrism. In studies of rats, animals fed a diet of 50% sweet pea seeds developed enlarged adrenals relative to control animals fed on edible peas. The main effect is thought to be on the formation of collagen. Symptoms are similar to those of scurvy and copper deficiency, which share the common feature of inhibiting proper formation of collagen fibrils. Seeds of the sweet pea contain beta-aminopropionitrile that prevents the cross-linking of collagen by inhibiting lysyl oxidase, leading to loose skin. Recent experiments have attempted to develop this chemical as a treatment to avoid disfiguring skin contractions after skin grafting. Genetics. Gregor Mendel is today recognized as the father of modern genetics for his work with the crossbreeding of pea plants with different characteristics, and sweet pea has been used in a similar way. The sweet pea is thus a model organism being used in early experimentations in genetics, particularly by the pioneer geneticist Reginald Punnett. It is highly suitable as a genetic subject because of its ability to self-pollinate and its easily observed Mendelian traits such as color, height and petal form. Many genetic principles were discovered or confirmed in this species. It was used by Punnett in early studies of genetic linkage. Complementary factor inheritance was also elucidated in sweet peas, from the cross of two pure-breeding white strains which gave rise to a blue hybrid, the blue color requiring two genes derived independently from the two white parents. Like the blue rose, the yellow sweet pea remains elusive, and a true yellow is unlikely ever to be achieved without genetic engineering. Gallery. See also, List of AGM sweet peas, List of sweet peas that have gained the Royal Horticultural Society's Award of Garden Merit. Notes, Euro Plus Med Plantbase, 1, C. Brickell, Encyclopedia of Garden Plants, 1996, Royal Horticultural Society, London, ISBN 0-7513-0436-0. Graham Rice, The Sweet Pea Book, Batsford 2002, pages 9, RHSAZ Encyclopedia of Garden Plants. United Kingdom, Dorling Kindersley 2008. PA 1136. ISBN A14053322964, Daster, D.K. and Ilya, C.G. Lathyrus vs. Odoratism. Nut. Rev 17, 33-6. Dorsler, W. Observations of Odoratism in the Rat. Journal of Nutrition 53, 105-13. Sweet peas make a second skin, Guardian, UK, July 2008, Punnett, R.C. Linkage in the sweet pea. Journal of Genetics 13, 101-123, Bateson, W., Saunders, E.R. and Punnett, R.C. Experimental Studies in the Physiology of Heredity. Reports to the Evolution Committee, Royal Society of London, 3.